I'm Alexander Kanaukhov. For many years I was working on WWF Russia, but before that I had been working closely with our today's speaker, with Dr. Miroslav at Savertsov Institute of Ecology and Evolution. Actually, Miroslav is still working there, and our Snow Leopard project was launched in 2010. Many camera traps were installed by our team in Tuva and Buryatia republics in the Russian side of Snow Leopard range. And also many scatter samples have been collected during these years. And it was Miroslav who was responsible and is still responsible for the further analysis of these samples. And what is especially important in this context, in context of today's discussion of possible migration corridors and connectivity between subpopulations, these samples were collected not only in the Russian side, but also in the neighboring part of Mongolia, of Kazakhstan, and as well as in other Central Asian countries. Actually, the results of this analysis form the basis of the article which we are going to discuss today. And now I would like to invite and give the floor to Miroslav. Over to you, my friend.
Hello, dear colleagues, dear organizers, everyone who has connected to the webinar, and grateful for this opportunity to have a lecture in this webinar. And it's a great honor for me. Okay, let's begin. Gene flow is vital for long-term viability and adaptability of species, preserving genetic diversity and resilience in changing environments. And functional connectivity among populations enables dispersal and migration, which is critical for species integrity and effective conservation. Therefore, understanding this connectivity is critical for effective conservation and of focal species, particularly the snow leopard. In the context of snow leopard conservation, studying these aspects is particularly important. First, given the high degree of natural fragmentation of its range, and second, the ever-increasing trend of habitat disturbance, reduction of the prey base, and population density of the snow leopards itself. In this context, genetic methods are invaluable in assessing functional population connectivity by evaluating population structure and in identifying genetic signatures of dispersal and migration. The snow leopard inhabits high mountain ecosystems of Central Asia, and its range divided into two main regions. This core or southwestern range, including Tibetan Plateau, Himalaya, Karakoram, Hindu Kush, Pamirs, and Tian Shan, and northern range, including Altai, Sayans, Khangai Mountains, and this range harbor an estimated 20–25% of global snow leopard population. Recent habitat and connectivity modeling studies underscored significant landscape isolation between the northern and southwestern areas of the range. Similarly, recent genetic studies revealed a pronounced genetic distinctiveness in the northern snow leopard habitat compared to other parts of the range. This division supports by both microsatellites and genome-wide data. Landscape models identified key potential corridors that may facilitate connectivity within the global snow leopard population. These include mountain ridge framing the Dzungarian basin in the northwestern fringe of the range as a potentially significant linkage between the Tian Shan and Altai, and the Gobi Desert and its surrounding range as an alternative linkage for connecting the global snow leopard population at the eastern edge of the range. These findings underscored essential areas that necessitated future survey efforts to gain a comprehensive understanding of snow leopard connectivity across the range. To date, only one gene flow study studying China and Mongolia indicates occasional admixture along a 1,000 Gobi-Singhai-Gansu corridor. Thus, the genetic data aligns with modeling predictions for a potential migration corridor in the eastern parts of the range. Meanwhile, the Dzungarian basin reaches east Borohoro, Dzungarian Alatau, Sauter Bogatai remain genetically unexamined despite their potential corridor role. Therefore, our study objective is to assess genetic connectivity through Kazakhstan habitats using mitochondrial DNA and microsatellites to detect historical and recent gene flow between western and northern snow leopard populations. In total, we used 93 samples collected across Russia, Mongolia, Kyrgyzstan, and Tajikistan. You can see these locations on the map. And among them, we analyzed 54 mtDNA sequences and 73 individual multilocus genotypes. The genetic markers we used were hypervariable region 1 of the mitochondrial DNA control region and 20 polymorphic microsatellites developed for snow leopards. Based on mitochondrial DNA analysis, we identified 7 distinct haplotypes, 5 of which were newly discovered. Within this fragment, we detected a repetitive sequence array, RS2, comprising 2 to 3 80 base pairs motifs. All Pamir samples and 3 samples from Tian Shan carried 3 RS2 repeats. The remaining sequences carried 2. The Bayesian maximum clade credibility tree indicated a division of haplotypes in two main clades. However, this division generally didn't correlate strongly with the geographical distribution of the species. And moreover, the maximum likelihood tree didn't recover the same subdivision. Similarly, the median joint network highlighted only 3 divergent haplotypes from the Pamirs and Tian Shan, which is distinguished by a third RS2 repeat, while revealing minimal divergence among the remaining mitochondrial lines. This outcome aligns with recent findings from a range-wide mtDNA analysis of the species, by emerging cancellara, which showed extensive overlap in the geographic distribution of maternal lineages, and furthermore, combined with the lack of mtDNA variation reported in a previous study by Jan Janchka, our data supports the notion of a weak phylogeographic signal is in snow leopards. Pairwise FST values indicate significant post-Pleistocene gene flow between Dzungarian Alatau and Tian Shan populations, which is not surprising given the high level of landscape connectivity in this region. However, due to statistical insignificance, the FST indices provide limited insights into genetic connectivity within the Dzungarian region, and the much higher FST value between the Dzungarian Alatau and Mongolian Russian populations compared to the Tian Shan may be an artifact of small sample size from the Dzungarian Alatau. However, these values could also suggest weak historical connectivity between Kazakh and northern habitats. The chain of potential habitat patches extending towards Altai includes the Taurus Bogatai ridges that might serve as stepping stones between the stable Dzungarian Alatau and Sayan populations. However, the Zaysan Basin represents a significant barrier to snow leopard movement, and in addition, the minimal FST between Tian Shan and Altai Sayan suggests that the principal historical movement route may have lain south of the Dzungarian Basin along the eastern Tian Shan spurs, Borohoro Range, linking southwestern and northern portions of the species range. One surprising finding from the mtDNA analysis was that snow leopards from the Tian Shan were genetically more similar to the Altai Sayan population than to those in the Pamirs, as indicated by FST values. This suggests historically more intense gene flow between the Tian Shan and Altai Sayan regions compared to the Pamirs. This finding aligns with a recent genome-wide SNP survey of snow leopards, which showed Xinjiang Tian Shan individuals clustering with Mongolian and Russian Altai specimens rather than with Tajikistan snow leopard. This can be due to that during last glacial maximum, the Pamirs, Altai, and the Himalaya Xinjiang mountains likely served as glacial refugia, while the Tian Shan was the post-glacial dispersal and admixture area of snow leopards from the Pamir and Altai habitat. In contrast to mtDNA analysis, Bayesian analysis of microsatellite data revealed a clear subdivision into two geographically distant genetic clusters, both by structure and gene length. However, both structure results and discriminant analysis of principal components show signs of admixture between the southwestern and northern snow leopard metapopulations. And this means that despite significant isolation, occasional contacts can occur. Evidence of this sporadic contacts may include the discovery of a genotype from the western cluster in a male snow leopard from the Mongolian Altai, and the genetic admixture found in several individuals from the Mongolian Altai and Gobi regions. The high genetic similarity between Dzungarian Altai and Tian Shan snow leopards has hindered our ability to identify the source population of this male. However, we can suggest potential corridors that might connect Tian Shan snow leopards to the Altai Sayan population. The first road could link the Dzungarian Electro to the Mongolian Altai through potential habitats including the Saurte Bogatai. This scenario is likely feasible considering the resident status of the snow leopard habitats on the Chinese side of Saurte Bogatai and the existence of small potential habitats in Xinjiang, which is crucial for population connectivity. Although there are no documented cases of snow leopard long-distance movements across the same basin, the possibility remains, considering reports of long-distance dispersal across the Gobi Desert and other unsuitable landscapes. A second potential migration route may proceed along the Borohoro range towards the southern Mongolian Altai and or Gobi, south of the Dzungarian Basin. Connectivity model likewise identified both northern via Saurte Bogatai and southern via Borohoro corridors as a likely area for population connectivity. Our structure analysis revealed greater admixture in the Mongolian Altai than in the Gobi, which may suggest higher northward connectivity through the Dzungarian Altai and Saurte Bogatai. However, another model found low landscape centrality of the Dzungarian Altai, but identified a linkage connecting the Tian Shan to the Mongolian Altai ridges south of the Dzungarian region. In addition, Hecker with co-authors in their modeling and genetic study demonstrated a high level of connectivity within the Gobi Altai and Mongolian Altai within Mongolia, alongside modest differentiation of Gobi snow leopards. Taken together, these findings may explain the elevated admixture observed in the Mongolian Altai relative to the Gobi, even when we account for potential migration through the southern Borohoro corridor. Thus, our genetic analysis confirms the Dzungarian region as a crucial area of connectivity between the species' northern and core ranges, thereby reinforcing findings from prior habitat and connectivity models. However, our current data cannot definitively determine whether Kazakhstan serves as the primary migration corridor in the focal region. Insights from our analysis, together with previous modeling and genetic studies, indicate that population connectivity in the Dzungarian region may also extend south of the Dzungarian Basin, particularly through the easternmost fringe of the Tian Shan range in Xinjiang. Interestingly, a fresh article on connectivity modeling present similar results. This study highlights more connectivity through southern corridor when considering empirical data, but also identified possible connectivity via Saur-Terbogatai, Kazakhstan, when using simulated data. Thus, we believe connectivity across the global snow leopard range relies on several narrow corridors, framing contact zones, facilitated by the species' capacity for long-distance movements. And recent genetic research confirms the existence of one such contact zone in the Gobi region. However, limited sampling in our study constrains our ability to pinpoint the most feasible gene flow routes, underscoring the critical need for additional data on Saur-Terbogatai range in both Kazakhstan and China, as well as the Boro-Koro range in Xinjiang, the Dzungarian Alatau, Mongolian Altai, and Gobi, which are essential for a more detailed and robust understanding of connectivity in the region. And given the Dzungarian region's potential to maintain gene flow between the two major parts of the snow leopard range, conservation priorities should focus on protecting habitats framing the Dzungarian basin. This includes the Terbogatai and Saur, Dzungarian Altao, and easternmost fringe of Tian Shan in Chinese Xinjiang. This would constitute important conservation measures to sustain or restore stable populations of snow leopards and their prey. That's so. Thank you for your attentions, and I would like to thank all my colleagues who are involved in our work, who make up our team. They are wonderful people, and I'm incredibly happy to be working alongside them. Thank you very much.
Miroslav, thank you very much for your presentation. Dear colleagues, you can ask Miroslav all what you want, or you can put a message in chat. We constantly talk about the isolation, especially of the northern snow leopard population, but as a rule, nothing happens beyond these words. The locations of possible corridors are very rarely surveyed, and in general, they receive unfairly little attention. However, understanding what is happening in these places is critically important for the conservation of snow leopard, especially in the northern part of its range, and in general for its conservation on a global scale. And today's presentation has just slightly revealed the secret of how the snow leopard's migration between the core of its population and the northern part of its range would have occurred. We have confirmed the critical importance of the Dzungarian Alatau, Terbogatai Ridge, South Altai Ridge for snow leopard conservation. These regions and their conservation undoubtedly deserve far greater attention. Now we must, so to speak, create conditions on the ground for unimpeded gene flow between Central Asia and the northern part of snow leopard range. And in addition, the natural isolation of the northern part of snow leopard range is enhanced by the development of a linear infrastructure, especially on the border between Mongolia and China. So it's a very big and complex problem. What should we solve to conserve snow leopard generally? And now we can discuss this article, this problem, and snow leopard conservation.
Thank you so much, Miroslav. That was a really interesting talk, and congratulations, a very interesting paper as well. So well done to you and your team. Sasha just mentioned a lot of the problems, obviously currently around transboundary efforts, with sort of the obstacles and barriers that are being sort of established that might limit dispersal. But actually, I think what I'm more interested in at the moment is just understanding more about the timing of the results that you observe. Can you give any indication about the sort of the rate at which we might be observing a fragmentation, and what that might offer in terms of the recovery rates, were we to get to a position where the range of snow leopards could be more contiguous?
Phil, will it be possible maybe to type your question? I think
Yes,
in
of
that
course.
case,
I'll type it in
to...
and then you can read it.
Thank you so much.
Wonderful.
Thank you so much. Thank you. Yes, I had a question. I was just waiting. Maybe once Phil's done, then I can ask my question also. Thank you, Phil. Now, so my question is, this is a brilliant body of work. I think it also validates some of the ongoing efforts being put in place. Like, I think UNEP, ILBIRS, and other organizations have recently identified a large corridor in the south of Kyrgyzstan. There is another project where Jeff and UNDP, in partnership with other organizations, are identifying and establishing corridors. My question is, do we have a slightly better understanding of what are the key barriers that should be looked into as we move forward towards blocking this gene flow? I know, I mean, some of them would be very standard answers like fences and all, but do we have some other parameter, like which parts of the distribution range are critical, in which case they should be very sensitively dealt with? And any observations on where the barriers come and where are those critical parts of the range which should be focused on when it comes to maintaining those corridors?
Okay, yes, thank you for the question. Я думаю, что глобальная фрагментация ареала прежде всего обусловлена естественными причинами, то есть разрывом как бы в подходящих местообитаниях. Вот. Но, конечно, снижение численности снежного барса приводит к более глубокой фрагментации, к нарушению миграций между популяциями. Ну, здесь разные причины могут быть. Это и сокращение численности самого снежного барса в результате браконьерства, нелегальной добычи, в результате, в первую очередь, наверное, даже снижения кормовой базы. Ну и, конечно, человеческой деятельности, таких, как Александр сказал, линейной инфраструктуры, разработок полезных ископаемых и прочего. Вот. Но как моделирование, так и наши генетические исследования, не только наши, но вообще, которые были проведены, показывают, что вот именно между северной частью ареала и основной частью ареала есть миграции. Они показали места, где действительно, которые наиболее важны для связанности популяции в пределах всего ареала. Вот. И наша задача сейчас это более глубоко изучить, какие именно используются коридоры или какие могут быть использованы или использовались ранее, чтобы сконцентрировать вот именно здесь усилия по охране. Ну, мне в остальном трудно сказать, какие еще могут быть препятствия для миграции, скорее, это вопрос к моделированию именно ландшафтной связности с учетом многих переменных. Вот, но, в общем, я думаю, так. Yes, I think the global fragmentation of the range is primarily due to natural causes, that is, a gap in suitable habitats. But of course, this decrease, the decrease in the number of snow leopard leads to deeper fragmentation, to disrupt migration between populations in global range, and different reasons can be this and the decline in number of the snow leopards itself, and also this can be decrease in prey base. I think this is maybe primarily problem. And of course, human activities, like Alexander said, linear infrastructure, developers to minerals and stuff. But I think modeling and our genetic studies only have been, and not only others that have been done at all, shows that there are that the main part of the snow leopard range is connected by migration, and they have shown the exact places where really that are most important for population connectivity within the entire range. That's our task now. Our main task now as geneticist is to study more deeply which corridors are used, or which can be used or have been used previously, historically. To concentrate here on the conservation efforts, and it's otherwise hard for me to say what other obstacles there might be to migration. Rather, it's a question, I think, of modeling exactly landscape connectivity, taking into account many variables here, but in general, I think so.
Thank you, Miroslav. And we also have a question from Philip Riordan. And from my opinion, it's a very important question. How much time does it take to see the changes in genetic relatedness across these regions? Сколько времени должно пройти, чтобы мы могли увидеть эти изменения на генетическом уровне? На основе наших данных сказать, как давно эти два участка популяции были соединены друг с другом и не были изолированы. То есть вопрос о времени.
Хорошо, well. Just a moment.
Miroslav told that it's difficult to say about timing based on our research, but his opinion that this isolation was during the last glaciation. Also, Miroslav is not ready to comment on all potential sites of gene flow, but as for the Dzungarian Altai Mountains, one can say that the isolation is natural, but we must make more efforts to increase connectivity now, because the snow leopard number is decreasing in eastern part of Kazakhstan, in whole Russia. So we need to make it possible to free migration for snow leopards.
Скажи, пожалуйста, Саш, если можно, вот про то, что вот статья Грачева по моделированию трансграничных коридоров, она как бы тоже показывает вот это, что как бы основная миграция сейчас, судя по всему, идет с юга, там через Борохоро, но исторически как бы она тоже потенциально могла идти через северный коридор, вот, когда численность была выше снежного барса.
Miroslav told that there is more information about possible gene flow corridors between northern part of snow leopard range and Central Asian one is in the publication which is on the screen.
Thank you. It's helpful to know.
So, dear colleague, for your attention.
Okay, thank you so much. It was a very great presentation, very insightful. Thank you so much for sharing with us. We're going to send everybody who sent the email the presentation. And feel free to send us any questions as well that we can forward also to our presenter. Thank you so much.
Thank you. I would like once again to thank everyone who who joined to this presentation, to this webinar, and we are open for any collaboration, for any questions. I don't know, via Alexander or by email. Thank you very much.
That's wonderful to hear. Thank you so much.
Thank you.